lihtne jääb kinni ja
Good morning to all the guests, presenters, and the people online. It's great to have you here on this typical rainy Estonian morning. My name is Siim Pers, and I'll be moderating the 2 following days. But enough of me, I'll invite here the principal of Tartu Jaan Poska Gymnasium, Mari Roostik, who will be opening the Poska Academy number 6.
Good morning, everyone. I heard you had an eventful and very fun evening last night, so the most important part of this conference is done. You've had fun and you've made friends. I will only take brief 4 minutes of your time, and I will start now. Start the clock, 4 minutes. When I was in primary school, I always looked at high school students as the most awesome people in the world. They're smart, witty, and they always seem to have so much fun. Now, when I'm a bit older, I still look at you guys as the most brilliant and awesome people in the world. Why is that? Because right now, you have so much knowledge in so many fields. Right now, at this moment, you have the possibility to choose your own way how you are going to change the world. I hope this conference will serve as an opportunity to get inspired on your path to change our world. And dear colleagues from everywhere, it's because of you that this conference is happening. And thank you for supporting and guiding your students. You already have changed the world. So let's start your conference and have a wonderful 2 days. Thank you.
The program of the conference is also online by scanning the QR codes on the walls, as you can see, or typing in the address of the website, which is underneath the QR code. There you'll also find the information on how to access the Wi-Fi. The conference is broadcasted live on the web. The link to the live broadcast is available in the conference booklet on the website and the Facebook page of Tartu Jaan Poska Gymnasium, and also on the screen. Please feel free to share the link with all your friends, families, and teacher. You are also welcome to ask questions after each presenter has finished their presentation. To do so, all you need to do is put up your hand and I'll come to you with a microphone. Or type in www.menti.com and the code you can see on the screen during each presentation. The code is also in the conference booklet. Questions through menti.com are very welcome from everyone watching online as well. But now, we will go on to our plenary presentation. The first speaker is Sulev Kuuse, head of a laboratory animal facility, Institute of Molecular and Cell Biology, University of Tartu. And he will tell us about studying biology toward biodiversity. The stage is yours.
Hello, good morning. It's nice to see a lot of you here, and let's start now. My working idea and working principles are connected with transgenic animals. After all, I have managed very much with students also. With students, we go in each year to the International Biology Olympiad. It means that I know quite well the situation at the school also. But here I am the first time now. And now let's go what I can say to you, and some moments. Yeah, it's better. It's nice. Okay, thank you. Let's go weiter. Here, I put here the first picture about one animal. What do you think? Which kind of animal it is? It's drawing by Gaius Plinius Secundus in, as you see, about 2,000 years ago. What kind of animal? What do you think? Yeah, they are egg, and it licks the eggs. And what kind of animal? It's bear. Bear, yeah, really. And in this time, 2,000 years ago, the people are thinking that the baby bears will be growing in that way. At first they will be here, and now after linking, they are in that side, and after that they can go in normal size also. It's quite interesting understanding about the whole world at that time. Now one picture about Salvatore Dali. Here, as you can see, a lot of stones, and when you look when you look closer, it's a woman here. It's also one way to understand the world. As I said before, that first we must have some imagination. After that, we must have culture in our word. It's Salvatore Dali. And now it's also art here. Here you can see it's made in 2005. They are our studying objects here. For example, it's the very small embryo of the mouse. It's stadium called morula. It's the embryo from chicken. And now the artist will give us different kind of pictures here, and it's quite interesting to see how scientists and how artists will be going in one way. It's quite nice, I think so. Okay, let's go to the science now. Here the science. This picture from Karl Ernst von Baer. Baer is connected also very much with Tartu. Baer, the last living years of famous embryologist Karl von Ernst von Baer was in Tartu. And it's the rule of Baer. As you can see, the first steps of the development are quite similar. Here it's the fish, and here human. Not very much different. But now we know, of course, that development of chicken or development of pig is quite different in the future steps. But first steps, they are quite similar. So let's go on. Now some words about laboratory animals. Laboratory animals are very important because without them we haven't no possibility to understand the human body also. We don't have experience to do experiments with human. It's the reason what we must have different experiments, different experiments with animals. And mostly we use in our institute. I am 500 meters from this place in our institute in Riia Street. You know the zoological museum there, and after that is our institute also. And there we study mostly mice, rats, but the most work, the main work for in our institute is connected with yeast cells. And when we have some results with yeast or some results with plants, then we must go only, after that we must go to animals. and animals, mainly mice. And this is one common house mouse, mus musculus in Latin, and this mus musculus is very useful. They are another species of mus musculus. It's also the common mouse, but it's without hair. It's called nude mice, nude mouse and nude mice. It means that they are in this mouse we have one mutation during the development of T cells. You know which are the T cells? Yeah, these, you know that, but you know also. T cells are the immune cells in our organism. And in T cells development, they have one mutation, and after that, they haven't no T cells in their organism, and they haven't also four. They called nude mice, and they are very important in our studying also. It's one common yellow-necked wood mouse. It's such name. It's also in natural species in Estonia and in Latvia and in Sweden and all Nordic countries also. It's not for using in labs because we never know which disease this mouse will have or what kind of condition for living they have and so on. It means that when we use mice in labs, it's very important to hold them in controlled situation. It means that the temperature must all time the same, the light and dark situation must be the same, and so on. The food must be the same. Now it's black or common rat. It's also, I don't know, in Estonia it's only in Setomaa region, not more, because here one interesting field in the nature, we know that, you know maybe also, that we have in Estonia two species of rats. Firstly, the common rats, and after that the Norwegian rat, or it means that I don't know, Rattus norvegicus, it's the name. It's a traveling rat. It's common translation of this species. And now is the situation so that the common rats population is very small because the Norwegian rat will be mating with common rats, and after that no pups will be born. It's the difference of two species, and after that is the situation so that only Norwegian rats will be breed and give the pups, but no black rats. And we must also study this species also. Here one common family of lab rats, lab mice, and as you see, not mostly in one family can be black rats or brown on that kind of gray rats, but it sometimes happens so that in one family is different mice with different colors. It means that in the nature we have all times some mutation, we have all times different conditions, and it's not clearly what's reason we must have in the nature that we when we want to get special result. It means that when we want to get only black rat mice, it's very not so easy to get them. Here one picture also about different mice. Here I want to go to some words to tell to you about transgenic transgenic animals. It's one mice. They are at the same age. They are about 1 month old. It's the brother of this mouse. It means that it's the color of this mouse is different. As you can see, this eyes is different. Here, the normal mice eyes can be black, or in some species also red. But here they are not so nice looking here. It means that here we have one example of genetically modified organism. It's the mouse which have some mutation in its organism. And now we must understand what causes this kind of eyes, for example. And here one picture about it's neat for to talk with this microphone. Yeah, it's so important. I want to walk here. It's maybe okay. Äkki jah. Ma võtan. Okay, now it's better. It's not so good to speak here. I want to say that here one animal, it's a rocky hyrax. It's from Serengeti. I got this picture, but it's not so common species in world because there are several conditions that are not good for this living, their living, and we want to save for other in future generation also that kind of animals. And for that we must study also all these distinct species and species which can be distinct also. Here one picture about UK. It's you can see here how many works with laboratory animals are done in world or in UK in these years. It's what's the year? 2018. Here you can see that in UK they used more than 2,000 mice, and from avian species about 50. It means that they use them for studying special processes and special interesting fields they want to study. And here you can see the level of severity of these experiments. They are not much influenced to the animals. Sub-threshold is the mean here. And you can see only 217.5 was in this level. But quite a lot of mice here have very serious severs here. You can see that more than 300 have injuries and so on. It means that we must do that kind of experiments. And these kind of experiments we can do only with lab animals. We have no possibility to understand that kind of processes. Here, one picture about Estonian experiments with lab animals. For example, here, compare with 2018 years. Here, it's better. We have in Estonia done experiments with approximately 2,000 mice also. Here, for example, 4 pigs have in the experiments, what more? 400 fishes and so on. And here, 1 point more, as you can see here, we have different GMO rat and mice, which we can use. It means that they are modified organisms, and they are very good to understand what happened when we have, for example, knock out one gene from organism. And we can do this only in the labs. Let's go wider. What we can do in, for example, in Estonia, what kind of experiments we can do? Here, basic studies, it means that we must study all basic situation, all basic injuries and all basic diseases of human, which we can introduce to the animal. And so it's impossible to do it only. After that, some medicine studies. It means that we must study how one medicine will be affect a person, and this kind of experiments we can do also only in animals, not in frogs or not in insects, but only in animals. We must study behavioral studies. It's connected with medicine studies. When we have taking some medicine, we must understand how it's connected with our behavior. It means that when our feeling after taking this medicine is not good, or we can't walk after that, maybe it's we can study with different behavioral experiments, and how it's have connection with human also. We can imagine it. So after that, some disease models. What it means? You know that in the bird we have different serious diseases, for example, different neurodegenerative diseases. For example, Parkinson, Huntington, Alzheimer. They are very serious diseases, and also without animals we can't study them. Some pictures about when we can catch this place, then I talk you more, maybe we have possibility. Transgenic animals, to get transgenic animals from studying of disease models. And one thing I mentioned here especially, you know what means the monoclonal antibodies, you know or not? They are that very specific type of proteins in our organism that can fight against different bacterium, different fungus, and so on. It's important to know immunology, and it's important to know that we have in our organism T cells and B cells. And B cells can produce monoclonal antibodies. And it's very specific field, and for that, when I remember right, in 1986, Cesar Melsten and Köhler got Nobel Prize also. It's very important field in our studies. So let's go on. Here some pictures about different experiments which we can do. Here you can see pups of mice. As you see, they are normal size pups. They are 7 days old. They can't see. They must drink milk from mother. But here you can see 2 small pups, and it means they can't get milk from mother. There are different reasons, but one reason is that in these mice they haven't one transcription factor for development, and it's influencing so that they can't eat. Here one picture, and here also a picture of about one tumor. It's lung tumor. You can see this the normal structure of lung, mice lung. And here, this the black side of lung. It means that there is the tumor cells growing. What do you think? Why they are black? And this the normal structure of the lung, but here why they are black? Have you some idea? It's a normal structure, but they are growing uncontrollable, and they can't give us the normal lung structure. They are cancer cells, and the reason is that here in the tumor must be very good energy and energy changes with mother. Well, not with mother, but with the normal organ. It means that there they have very much of capillaries, And this gives the structure that they have black. In capillaries, the blood, and blood is, when we took from our organism, it's very dark. And now when we have here very much of capillaries, it's sees that it's the dark places. And here one cancer also, it's epithelial cancer in the between the fur of the mouse. In the world, how many time I have? How many time? What do you think? 30 minutes more. Yeah. Okay. First GMOs in the world. They was Escherichia coli. You know, it's the small, not pathogen. It's in our organism. And now in 1973. They put in the Escherichia coli some Salmonella species exogenes. It means that they have made not common Escherichia coli, but it's have some genes from Salmonella. It's different species. And after that, 2 years later, in the world was the Asimolar conference. It's a very important conference that time. It's more than about 50 years ago, and they told there it's easy, it's a very risky business, or it's normal to study that kind of experiments, to study different organisms, and to study organisms which we have made change in different ways. And after that, as you see, 3 years later, it's E is absent here. 3 years later, we have in studies some cells which can produce more insulin. Insulin is very useful medicine in our world. In humankind, it's very important to have this insulin more for treating illness people. It means that 3 years later already we have some results. It's not nice picture, I think so. It's not must be here. I have forgot this picture here. But now this also one very, very interesting field in GMO world. It means that it's synthetic cell. And what it means, in year, not approximately 20 years ago, one person, Craig Venter, studied this mycoplasma, it's bacterium, and he have an idea that he have done this experiment that way. From mycoplasma cell, it's not the same cell, but from mycoplasma cell, he removed all the nuclear material. It means all DNA was removed from that cell. And after that, he synthesized by computer and by machine new DNA and put this new DNA to these cells. It means that it's the Craig Venter here, and he got at first Cynthia. That is the name of this Cynthia Craig Venter Institute. It's the name here, and he got Cynthia #1. After that, and this project was quite expensive, as you see, approximately $40 billion. Now he continued this work, and now he have results from year 2016. He got Cynthia free, and now we know that it's this Cynthia cells, this is a Cynthia free, it needs only 473 genes. They worked, and these cells can divide, and they can live. Now one question, how many genes we have, you know? A lot, it's right, a lot. Yeah. Approximately 10 years ago, it was thinking that we have more than 25,000 genes. But today, some weeks ago, was some information from scientific community that we have only 19,000 different genes. They are functional genes. After that, we have, of course, non-functional genes and so on, so-called housekeeping genes. But here in human, we have 90,000 or 20,000, but here for living is enough from 500 genes. It's also interesting to understand. Of course, we can't compare this kind of cells and human, but some point of thinking they have for us. Here one example of plants, transgenic plants. It's also important, you know that in the world which have war places, for example in Ukraine, but in different places we have also war, and there we need to use different explosive components. And in US, in one military factory, they have prepared different bombs and different explosive agencies, and there is problem that some things of the factories goes to the water, and from water they go to the soil of the US, some region. And there, what to do then? The soil was at all destroyed, no plantation there was, and so on. And they used one type of very small flower. It's in Estonia, it's murloog, but I don't, weed in Estonia, in English, weed species. And it's a very common species of studying development in the plants. And now they use that kind of GMO weeds, which it's the name Arabidopsis thaliana. And these kind of plants, they are quite small, 10 centimeters long, I think so. It's the one part of this here, the, not the uterus, but in the plants what it is. You know, maybe. I don't remember this word in the English. Sorry. But here you can see this, it's confocal microscope picture about this plant flower. And here you can see different parts colored with different fluorescent dyeing. But the most, what I want to say, that they used that kind of technically modified weed species, and this species have possibility to destroy these chemical agencies. And after that, the soil was also, with the soil was all okay. After, I think after 5 or 10 years. It's quite a long time, but it's better than nothing. So we can, what happened now? Yeah, so thank you. Here we can see that that we have different possibility to study these processes. It's transgenic, chimeric, mosaic, and hybridic. It means that with DNA is something wrong or something is happened. Transgenic, you know that, for example, they are these mycoplasma cells. Transgenic means that into these cells we can place different genes from other species, or from other taxons of other organisms. Transgenic, chimeric here also, it means that here this is the wild type mouse, it means this is the common mouse, but when we have changed there some genes, they are not changing in all cells, but especially in these cells, the full color shows us, in these cells they these there we have some changes into the genome. And these brown cells, they are wild-type cells. Here we can also talk about mosaic organism. It means that some Ebola viruses and some different viruses, for example, this coronavirus also, they can move and change its genetic consistence freely, and it's the reason that we can't fight against them so easy. And here, one possibility more, it's hybridic organism. It's so-called collar bear, or grizzly bear, or nanulak. It's different in different languages. It means it's the polar bear which have mated with grizzly bear. And after that, we can get that kind of species. They are very, very angry. They are very, very awful for human also. So it's normally happens in the wild also. So let's go on. Here are some pictures about transgenic also. This is the common mold. One common mold species, they are here you can see this is the normal. Here it's not, so it's there is no modification. But here you can see some mouse embryos. Mice embryos. Here one embryo. It's the normal developmental embryo. Here this embryo is about 8 days old after mating. It means 8 days old, and now when you want, at older age, when it's burned, this embryo, and now we have living mouse, then we can see that the renal structure is quite normal. It's the renal, and this point means that in this ren we have one specific gene. It's called kata6, and it's working here normally. But when we have, for example, it's quite interesting system for studying transgenic animals. It means that it's also transgenic, but here these transgenes can't no influence for the organism. But when we have here, it's tox, it means that it's one antibiotic toxocycline. And now when we add in the drinking water for the mice this toxocycline, it's all normal. But when we remove this toxocycline from drinking water, these mice don't grow normally. As you can see, the renal structure is quite different. They are not so quite nice structure here. The point of kata6 gene, they absent here. And here you can see also the embryo. It's the embryo development is also not normal. And that kind of experience we can do, and in that field we can understand what happened with different organisms. Here, for example, it's the urinary tract. It's urinary tract development, and now here it's the normal situation. But when you see this point, here this point, and this point is here, you can see some signal, but here no signal. It means that in this kind of genetically modified mice, no urinary tract development we don't see. It's one common genetically modified mouse, and in the EU community we have from 1990 also that kind of regulation, and it's quite important to have that kind of regulation, because without them we don't never what can be happened in. It's maybe not. It's not fun, I think so. We never know what will be happen in our science community also. When you know that 3 years ago in China have burned two child, they are knocked out from HIV receptor. It means that they can live, but this research staff have not allowed to do that kind of experiments. And after that, then it was quite big discussion in scientific community after that experiments, and now we know that leader of this company or this research group have 3 years in prison now. It's quite difficult to, without these rules, we can't do nothing. We must have rules. I can say so. Here are some pictures about different organisms. For example, Cnidaria, one very big phylum in the world sea, and now it's also genetically modified species of that phylum, and you can see the gene fluorescence here. Normally, you don't have that, but here, when the genes are modified, we can see that. Some transgenic insects are also important into the in world. It means that this region of the world, where for example, is spreading malaria, for example, or different illnesses, for example, dengue fever, or they are more. We have insects which will leave, I don't remember again the right name, but they can spread this virus from one species to other species. But here, at first, one picture about pink bollworm. It's also one big problem that the bollworm seed from cotton farming, in cotton farming was damaged when this population is very big. And now the scientific community have made that kind of bollworm which don't have offsprings. It means that they are genetically modified in that way that they don't have offsprings. It's for the cotton. It's cotton plants. It's very good, but we don't know what happens with different birds. What happens with different insects when we modified one species of our world? It's quite difficult question, and it's not so easy to answer this question. Here it is the pink bollworm, and you now see how this lies its eggs here, and after that we have here that kind of situation that no products we can't get. It's now in our institute also we'll study different organisms after mice and after bacterium and after yeast. We can study also Drosophila. It's small flies, and they are very useful for studying because they have very understandable, very well-known genetic information. DNA consists, and it's available from 1987 already. Now one picture. Let's see how I can open them. Please, the first one. Here, one picture. You can see here some pink dots. Here, for example. No, it's not. Let's go to the other video. Maybe it's better. Yeah. It's better to see here the cells, and now you can see the dots. It's live imaging of cells of Drosophila. They are wing cells of Drosophila, and the author of this videos was Osamu Shimi. He is working in our institute also. It's Japanese guy, and it's from Finland. It's a very national community in our institute also. And here you can see that how will the cell structure moved and how they will how they have different places to the development, different places to see how processes will be going on. So let's go weiter. Here, for example, with genetically modified methods, we can get also some pets in our world. They are zebrafish. Zebrafish, they are very useful objects in the scientific community because they are very fast growing, and we can study, we know this genome and we can study them very well. And now from year 23, they are patenting as the first living organism in the world. It's also interesting. Maybe you know that we can't patent no human organ or new human developmental things or no genome and so on. But here, one example, in 23, we have patented one living organism, and it is zebrafish. Quite similar species is also Medaka. It's Japan rice fish. It's also used very much in the laboratory studying. Here one maybe it's maybe better example for studying in the world about genetically modified fish or animals. It's the common salar here. Common salar, which lives in near the coast of Canada. And no, here some other places also, but they will study it there at first. Now it was so that from this fish, Salmo salar, where the scientists took from this fish one gene. It's they got from this gene, and they mixed this gene with ocean boat. It's one species also in the deep water, and the ocean boat have so-called antifreeze protein gene. And they mixed this antifreeze protein gene with gene of Atlantic salmon growth factor gene, and after that, these salmons will grow very fast. When we have in the market this big salmon, it's like so, 50 centimeters long, but for in the manufactory, we need, I think, that 5 years to growing in that side. But when we have done this kind of experiments here, this also the Salmo salar, but it's modified with modified genes. And now this is growing much bigger, and it's growing much faster also. It's for feeding of for human. It's very important to have that kind of speed of the getting of normal feed for eat table of our people also. And now it's the first submitting of salmon to do experiments was in 1996, but it's allowed it to use by human only after 20 years. It's allowed it, when I remember rightly, it's allowed it 2019. So it's one example how we can use the genetically modified species also. It's about birds, but now some words about how many time I have? 10 minutes? 5 minutes, okay. Will be shortly. What we need to do with animals. Here are some examples. Here, genetically modified mice. You can see this fluorescent point here, for example, nose, tail, and ears. They are, tips also, they are colored here. It means that in this organism we have different foreign genes. Here one very important example about marmosets. They are small half monkeys in East. They mostly are living in Tallinn Zoo. They also, you can see when we go to entrance of Tallinn Zoo, there one big cage from this kind of marmosets. And now it's also genetically modified. For us, not allowed it to do experiments with human. It's not allowed it to do experiments with primates also, for example, orangutan or chimpanzee. It's not allowed it at all. Only in very few points of interesting or very few points of science we can use, for example, pavians. They live, the most closest place to the Estonia is, I know, in France, in Lyon. There they have one big lab animal facility for growing pavians. And now to study the nearest species for human, it's also, you can see this the marmosets. And we can study these experiments, we can study different genes and so on. It's one genetically modified cat here. Here it's you know, maybe we have in human HIV, but in cat we have FIV. It means the Felis immunodeficit virus. And now here they studied this cat with this, and they studied how to treat this cat with different medicines. Here, one picture also about that kind of Belgian blue bovine. It's not so nice picture, but it means that in nature it's not very often, but sometimes it happens so that during the normal mutation, it's normal mutation for that cow, and it's the muscle mass is 3 times bigger or like compared with normal cattle, normal bovine. And here, as you can see, Salvatore Dali also was one scientist who, I can say scientist, artist, who can do some experiments with genetically modified organisms. It's imagination about yourself. Okay, I think let's I see. Oh, okay, they are not very good picture here. Next time, I will continue. Have you maybe some question? Yeah.
What do you think about legislation with the genetic modified organisms? Do you think that legislation will be able to keep up or even regulate the science and development?
Of course, it's very important, the legislation of this kind of experiments. I also taken part in one legislation company in Estonia, and I know very well this situation. 50 years ago, we don't need nothing to say to principals or other people. We only have done experiments. But now, when we want to do some experiments, we must have before different licenses to do this kind of experiments, and it's very important. I know that in Sweden it's also very important field, the legislation of that kind of experiments, and in Estonia also. Now it's very common and very important also.
So there is one question sent in through the internet. So what do you think about countries like China that make genetic changes to embryos, especially CRISPR-Cas9?
Yeah, CRISPR-Cas9 is one method in the changing field of DNA, but it's not allowed it by human. It's at all, it's not allowed. I don't like this kind of experiments. I have here, after it, I told you that in next time we must talk this discussion. I have here some pictures also about these experiments which made in China with human, and it's not allowed. Yeah.
The salmon that was genetically modified, you know, the taste modified too. Was it good for sushi?
Yeah, for sushi it's maybe good, but at first you must go to Japan and there to taste the sushi. I think so. But I can say so that it's better to live human so we are now. We don't need to move our genes. We don't need to do experiments with human, but we don't know these processes which have done in which we can done in other organisms, for example with mice or with insects. It gives us the information how we can do it and how we can prepare some mistakes which will be happen spontaneously. Thank you. Have you some question more? No? Thank you. And next time we can meet.
I'm really sorry that the microphone today is a bit moody. If you like, I can give you the wireless microphone, but it's better if we use this one. I'm really sorry, technology world we live in. I'll try to fix it. Poska Academy number 6 wouldn't be possible without the funding of Norplus Junior and the Tartu City government. Our cooperation partners are also student companies Vilput, Batoonipoisid and KOV. Now I'd like to give the floor to our cooperation partners who will introduce their products to you. All our guests are presented, all our guests and presenters will find their products in small gift packages waiting for you. Unfortunately, the representatives of the student company Vilput couldn't be here today, but I'll gladly introduce their company. Student company Vilput produces card holders from birch veneer leftovers. The convenient card holder boasts a unique design and can hold up to 4 cards, but 1 or 2 cards won't fall out either. The card holders also have a RFID system that makes it impossible to use the card through the pocket, preventing accidental card payments. The holder has been specifically designed for Poska Academy. I'll show it as well. It looks like this. Now we know about Vilput, but Sander Saale and Paul Supping will tell us about their student company Batoonipoisid. Kuidas endal mugavam on?
Hello, we are the student company called Batoonipoisid. Our logo is in the middle right here. So, hello again. We are the student company called Batoonipoisid. Me and Paul and 2 others also couldn't be here. Yeah, so pretty much that's the logo in the middle is ours.
So, for the last 9 months, we have been evolving and producing this product to sell it on the stands at the bigger supermarket, well, not supermarkets, but shopping centers in Tartu and Tallinn. And also, we have sold this in our school, and the feedback we got was really positive for almost everyone.
You might wonder, why do we talk so highly about our product? It is because it's made out of natural ingredients such as berries, mixed fruits, honey, chocolates, and different kind of nuts. There are no synthetic ingredients in here, so it's very healthy for the body, and it's easy to digest, therefore giving you fast energy from a healthy option. You all probably will get one, so let us know if you like it or not. Thank you and enjoy.
Well, looks like the microphone has folded again. I'll try to fix it once again. I promised you yesterday it held really well. Well, let's hope it works for now. Also, the student company KOF, you will meet them in person in the third session, but you can already taste their products, the plant-based coffee cream, during the first coffee break. Now, our conference will continue with 3 student presentations that will be commented on by the 3 of our friendly members of the Defense Committee. Let me introduce them to you. Master's degree coordinator at the Department of Science Communication, Estonian Research Council, Mare Vahtre. PhD, Associate Professor of the German Philology, Head of Department of German Studies, University of Tartu, Reet Bender. And last but not least, our school's alumna and medical student at the University of Tartu, Anni-Britta Pajomaa. I have a lovely bell on my table, and I will ring it when either 5 or 10 minutes is over. This is a notice for the presenter or presenters that the time is up. If you still have some information to give, finish it up in a minute. When the time for question is over, I'll make a remark on this as well. But now, let's welcome the first brave participant, Tuve Gabrielsson from Tivoli Gymnasium, who will present us a topic with which many of us won't be familiar with, detection of ovalbumin in lyophilized milk extracellular vehicles for immunotherapy. The stage is yours.
Hello, my name is Tova, and I had the opportunity to do a project with the Karolinska Institute in Stockholm, where I worked with a group that is trying to find a cure for allergies. So they are trying to do this by loading allergens into extracellular vesicles from milk and then giving them to the patient. So this is a picture of an extracellular vesicles coming out from the cell here. So they are also called EVs for short, which is the term I'll mostly be referring them to. And they are a group of very small particles, and they include exosomes and microvesicles. As you can see in this picture, we have exosomes here on the bottom and a microvesicle on the top. So they are produced from cells. Microvesicles budding up from the cell membrane and exosomes coming from inside of the cell. Their main function is cell-to-cell communication, yet they're still being researched a lot because they are quite newly discovered, so we're not quite sure what their full purpose is. But we know that they are used for communication, and they are typically about 100 nanometers in size. In 2005, EVs were found in breast milk, and scientists believe that they actually protect children from becoming allergic to food allergens. This is because the food that the mother eats will have their allergens present in the EVs and will therefore be transported to the children. So therefore, we believe that milk EVs could be used for curing allergies if the allergen of interest is loaded into the EVs. To make this possible, though, we need an efficient way to load allergens into the EVs. And for this, we're using a newly discovered process called lyophilization. It's a freeze-drying process that works by destroying the extracellular vesicles by the freeze-drying and then loading the allergen and then letting them reassemble with the allergen inside. This method has not previously been tested for milk EVs, which is what we're doing in this experiment. So my assignment was therefore to analyze the samples of the freeze-dried milk of EVs and investigate if the antigen loading was successful or not. And in my trials, we used ovalbumin as a substitute for the allergens. The results that we got was that we found ovalbumin to be present, as shown in the first picture. As you can see, you have a clear line at about, in the second column, which was the lyophilized EVs. So we can see that we did find ovalbumin. And in the second picture, we can see CD9, which was the other antigen that we tested because it is very commonly occurring in EVs. And we did, this test was inconclusive. As you can see, the entire membrane is colored in, which was not supposed to happen, so we would need to find a new protein to repeat this project. But the most important thing for this experiment was that we saw ovalbumin. So what I did was I performed a protein measurement as well as Western blot, which is, the Western blot is what we can see in the pictures. So this results, this suggests that the freeze drying was successful because we could load ovalbumin into the EVs. And the next step would then be to add them to the immune cells to further understand if this method could be used for curing allergies. And hopefully in about 5 years' time, we would be able to test this in patients and hopefully cure allergies. Thank you.
I'll bring you a microphone as well.
Thank you, very nice presentation. I have one question. You know how it's regulated, the transport of E vesicles, extravesicular vesicles? How it's regulated, the transport?
The transport of? Sorry?
From cell to outside.
Cell to cell. Well, I mean, they bud up from the one cell, and then they can be absorbed by another. So that's how, because the extracellular vesicles, they include DNA or RNA, so they include this like genetic information, which is absorbed by the other cell.
Mm-hmm. You know, maybe there is some percent of transport regulated. It's called regulated transport of vesicles. You know something about this, or?
No.
Okay.
I'm not
Okay.
sure.
It was a very nice presentation. Thank you.
Thank you.
Thank you very much. Tack så mycket. You answered all my questions I had when I previously read your abstract here. But maybe just one question about, I don't know if you know that, but why milk extracellular vesicles? What are the pros here?
Yes. Milk EVs, this is because the milk EVs have other qualities than other EVs. Since they come from milk, they are meant to be immunosuppressants because their main purpose is to make sure the babies aren't allergic to everything, so they get the allergens from the mother to make them see that this is not a dangerous food allergen. So this is why they have special qualities, just the milk extracellular vesicles that others don't. So that's why we need them, and we can't just use any.
But did you extract them from cow's milk, or
yes,
there's
cow's
no difference?
milk.
Okay, okay, and they work as
Precisely.
good as
Yes.
breast milk ones. Okay,
Exactly.
thank you.
Thank you.
Thank you for the work. I do have one question. Are there any differences in different milks? As you said, you used cow's milk. What about human's milk or goat's milk?
Well, we do think that we could be using cow's milk for immunotherapy, but I would suppose that there are quite some differences between cows and human milk, for example. But I know that this group does a lot of tests on both. They collect both, and I'm not totally sure what the differences are, but yeah, I know that they research both, and we think that we could be using even cow's milk, because it would be quite complicated to collect human milk for immunotherapy. Yeah.
Do we have any more questions, comments? No? Then thank you.
Thank you.
Hello, dear listeners. My name is Josep Press, and the topic of my research project is building a parabolic dish antenna for receiving pictures from weather satellites. Yeah. So the aim of my practical research project was to build a feed for the parabolic dish antenna to receive pictures from weather satellites and to receive pictures from geostationary satellites and obtain new knowledge about the subject. So in the theoretical part, I give a brief overview of the radio communication, how it's used, and what are the main types of it. Then a brief overview of satellites and satellite communication. So, for example, what is GPS, what are weather satellites, and so on. And some overview of antennas, mainly parabolic antennas and dipole antennas, as they are mainly used in satellite communication. In the practical work, I show how I built the feed for the parabolic dish and the mounts for it, and also explain how the filters and the SDR, or the software-defined radio, works. So in the left picture, you can see the black box is the filter and the amplifier combo, and on the bottom you can see the SDR, or the software-defined radio. That is the part that converts radio frequencies to signals that are understandable by the computer. And on the right picture, you can see the feed, which is basically just a cutlery holder with a copper wire inside of it, and that is the part that receives the radio frequencies. And then on the left picture, you can see the stand I made for the dish. This needs to be quite big and strong, so it's welded from steel pipes. On the center picture, you can see the dish in action, or it's pointed to the satellite and it's receiving the picture. And on the right, you can see the dish with the mount on it and the feed, and yeah. So results. All of the set codes were achieved. I successfully received pictures from 3 satellites, which were the Russian Electro L3 geostationary satellite, then the Chinese Fengyun 2H, and an US satellite called EWS-G1. So here you can see the pictures. The left picture is the picture sent straight from the satellite, and it's in black and white because it saves a lot of data, so they can transmit the pictures quicker. And on the right you can see the same picture, but it's colored in post, so it's more appealing to the eye. And here are two similar pictures. They all pretty much look the same because the satellites are pretty close to each other, and so yeah. But on the left, you can see a picture from the Russian Electro L3 satellite. It's a bit brighter than the others, but it's very similar. And on the right, you can see a colored picture from the US satellite EWS-G1 or GOES-13. Conclusions. This project is doable with readily available materials from hardware store and so on. It needs some other things, like the SDR and the filters that aren't viable from regular stores, but they are also readily available. And also that precision is very important, and it's very important to cut or measure twice before cutting to not make any mistakes. And ideas for students in the future are that they should maybe experiment with higher frequencies as the measurements and everything becomes even more important there. And also one idea is to build a tracker for the antenna to not only capture pictures and signals from geostationary satellites, but also from high and low Earth orbit satellites, which may result in more interesting pictures. For example, pictures of the sun or so on. Yeah, that's all. Thank you.
So I'm going to ask the opponents questions from you. Was all of the building, including welding, done by you, the author?
Yes, I did all of the welding and other building myself.
What was the biggest discovery for the author during the building process?
The biggest discovery was maybe that it was a little bit easier than I thought, because I was thinking that it truly needs to be a really complicated process, but actually if you get all the measurements right and so on, then it's not too hard. Complicated.
And is the author planning to continue working on the subject in the future?
Well, I haven't decided for sure yet, but I think I'm gonna be trying to do similar things in the future too. Thank you.
Yes, thank you for your presentation and for your work. I have a simple information question. Is it possible to receive with such kind of antenna information from all kind of satellites, from military satellites, for example?
Well, different satellites use different frequencies for data transmission, and the more interesting things are on higher frequencies. In this project I used satellites which had the downlink on 1.8 gigahertz, but the more interesting things are on like 10 gigahertz and so on. And also the military satellites are, the signals are encrypted, so it's not, you basically cannot receive the signals without some illegal things. But these satellites are not encrypted. They are basically everyone who has the appropriate antenna can receive them. So it's totally legal and easily doable. Thank you. Yeah.
You take your microphone as well.
Okay.
You say that you get it from the satellite with the knowledge now about alternative facts, etc. And can you trust the facts that comes from the satellite?
I'm not sure I understood the question completely.
You said there were Russian satellites.
Yes.
If they send out from Russian satellite, there will be sun here today. Can we trust it?
Yes, I think we can trust it.
Okay. Second question, the dish, did you have to polish to get a good good transformation into your electronic equipment? Did you polish the dish, or what did you do?
Well, the dish had sat outside for I think like 10 years or something, so it was a bit grassy and dirty. So I just pressure washed it, and it worked good.
Okay.
The dish quality is not actually so important on these lower frequencies, and the dish can basically be full of holes too. Some dishes are, it just makes them lighter, but yeah, I just for good measure I washed it, and yeah.
Unfortunately, we don't have no more time for questions, so thank you, Joseph. Not only does Jurmala have a waterfall, they also have a beautiful coast with a lot of different species in it. Teresa Prinadola will guide us on the coast of Jurmala and she tells us about monitoring of washed up mollusks on the coast of Jurmala. The stage is yours.
Hello, I am Teresa and I'm from Latvia, and I developed a research project monitoring of washed up mollusks on the coast of Jurmala. And the aim of the study was to explore the coast of Jurmala and to determine the species of bivalve and snails washed up on it, and to perform monitoring. There were 3 objectives of this work: to study the available scientific literature on mollusk type species found in Latvia, and to inspect the coast of Jurmala and determine the classification of the washed up mollusk species, and to map the locations of the washed up mollusk species. Mollusks form a broad group of invertebrates. Most of the mollusk species are aquatic animals, and around 130,000 species are known worldwide. The Latvian mollusk fauna includes 173 species. Bivalve mollusks need water to live, and this class includes mollusks with 2 hinged parts. Among snail species, there is the greatest diversity in body structure and lifestyle. Snails are characterized by an asymmetrical body. There were 5 parts of this research project: analysis of the scientific literature about mollusk type species, sublittoral survey, identification of the washed up mollusk species, mapping the finds, and analysis of the results. Results of this research are that out of 173 mollusk type species present in Latvia, 10 species were found and identified. 6 species of bivalve mollusks have been identified, 4 species of marine and 2 species of freshwater mollusks. 4 species of snail, of which 3 species of freshwater snail and single species of terrestrial snail. And in conclusion, freshwater and terrestrial mollusk species are most likely to have entered the Gulf of Riga through River Lielupe, as well as through pipes that are installed on the beach, through which uncontaminated rainwater and groundwater are being floated into the Baltic Sea. During of the study, 1 species of bivalve cannot be identified, and now, with the help of Latvian scientists, these bivalve species have been identified, which had not been spotted in Latvia until now. In cooperation with Latvian scientists, the occurrence of this bivalve will be studied. Thank you.
You already know what I'm going to say. It's time for questions and comments.
Paldies, Teresa. I have a question about how do you differentiate the mollusks? If I were to go to the coast of Jurmala right now, or a bit a better time, maybe a summertime, what are like the key points or like the landmarks if you find the mollusk? Do you just look at the appearance? Do you open them up? What do you look for?
The appearance and size of it. I was using one website where there are all of the descriptions of the mollusks that are found in Latvia, and I also used a book where there are descriptions, and it was pretty easy for some species, but for others it was not so easy, and I did it with my teacher. Yeah.
Okay. And you said you found a new species of mollusk. Did you get to name the one after you?
No. No, it's that species lives in. It's not like till now. It's not have been seen in Latvia only, but yeah.
Great job.
Yes, thank you.
Yes, thank you so much. My question is, did you have had some previous studies to compare your results with them? Were there more or less mollusks as in previous times?
I tried to search for those research projects, but there wasn't any about Latvian coastline, but about Polish, and it didn't help me at all. So
yeah. Do you have any theory from where these new species, where it came from?
Like the climate is changing and it's becoming warmer, and yeah. But the scientist that I'm working with, he said that this species have been spotted in Estonia, and now it's in Latvia. So yeah, if it's in Estonia, it's like logical that it needs to be in Latvia too.
Do we have any more questions? Comments? If not, thank you, Teresa.
Yes, thank you.
So it's time to start with the second session. I will remind you some of the basics of our conference. The program of the conference is available online by scanning the QR codes. The conference is also broadcasted live on the web. The link to the live broadcast is available in the conference booklet on the website and Facebook page of Tartu Jaan Poska Gymnasium and also on the screen. Please feel free to share the link with all your friends, family and teachers. Also, I have this lovely bell on my table. I will ring it when either 5 or 10 minutes is over. This is a notice for the presenter or the presenters that the time is up. If you still have some information to give us, finish it up in a minute. When the time for question is over, I'll make a remark on this as well. Also, you can ask questions by raising your hand and I will give you a microphone, or in the internet in menti.com, and also the code is visible on the screen. Okay, but let's kick it off. The food was great, we are feeling good, and according to my maths, it's time to welcome Anette Arrak and Kadri Pahtama from Jaan Poska Gymnasium, and they will tell their story on mathematical tests for 10th grade on Kahoot and Socrative platforms. The stage is yours.
Was to make mathematical tests for 10th grade in Socrative and Kahoot platforms. So basically we had 3 main purposes. The first one was to make learning more enjoyable and diverse. Because teachers don't usually use online tests, and it would be different and therefore more interesting. The second one was to revise study the topics. It will give the students feedback how well they know different subjects and do they remember them well. And the last one was to give feedback to teachers and students, so teachers get an overview of the most common mistakes and which topics were more difficult, and students can know how well they have learned the topic and what they need to revise more before the exam week. In the theoretical part, we introduced the national curriculum for mathematics. We talked about what courses 10th grade has, and we also introduced Kahoot and Socrative platforms. We explained the different options when making tests and also the different playing modes. So Socrative has 3 different question types: multiple choice, true or false, and short answer.
So as seen, Kahoot has way more question types. We personally use the first 4 ones. Quiz, which is where 4 different answer choices are given and you have to choose one. Then there's true or false, where you'll have to choose if the statement is correct or not. And then with the premium version, there was a puzzle where students have to put the answers in the correct order. And lastly, we used write, where you type the answer out.
In Socrative, there's 2 versions to carry out tests. First one is quiz, which is a typical test version, and the second one is space race.
which is same as quiz, but it's like a competition. The exit ticket is for asking feedback on the lesson. Also, there are some settings for carrying out tests. For example, you can set the time limit, and also there are 3 ways to carry out tests. Open navigation, where students can move back and forth between questions. Teacher paced, where the teacher controls the flow of the game or test. And lastly, instant feedback, where the students can move only forward. In Kahoot, there are 2 main ones. There's slides, where the teacher can make a whole course, and where they'll be able to put on slides and introduce a new topic. And the second version is Kahoot Teach or Assign. So Teach is the most traditional one, where you'll show the answers and questions on a big screen. And Assign, in Assign version, a student can see the questions and answers in their phones. And in Teach, there's a time limit from 5 seconds to 4 minutes, and it can also be applied to Assign, but it doesn't have to. And there's also multi-select, which means students will have to choose, in quiz for example, they have to choose more than one correct answer. And after each question, students see the leaderboard of how well they're doing. Both in Socrative and Kahoot, the teacher sees in real time how the students are doing, and after they test, they'll be able to see more specifically on which student or how one specific student did, or which questions were the hardest. And also in Socrative, the results can be downloaded as an Excel file, and in Kahoot, there's automatically statistics compiled. We made math tests for each 10th grade course. First, we came up with different questions in our shared Google Docs file, and after our supervisor looked them through and gave us the green light, we put them in Kahoot and Socrative platforms. I put them in Socrative, while Kadri put them in Kahoot. The first cycle was about expressions and sets of numbers. Socrative had 2 tests, while Kahoot had 3. And also the numbers in the brackets are show the number each test had. The second cycle was about equations and equation systems, and both platforms had 1 test each. And the third cycle was about inequalities and trigonometry 1, where Socrative had 1 test and Kahoot had 2. The fourth cycle was about trigonometry 2, and both platforms had 1 test each. And the last cycle was about vectors and equation of a line, and also both platforms had 1 test each. On the 15th of November in 2021, we did a pilot experiment with class 10C. We made them 3 analogous tests on equations with exponents and square root. And the tests were different. One of them was Kahoot Assign, another one was Kahoot Teach, and the third one was in Socrative. And after that, they filled the feedback form, where we found out that 85% of the 32 students who answered the feedback found that the tests support studying. 50% of them prefer to have the tests in the middle of the cycle. About a third of them would like to have them before the exam week. Most favored one was Kahoot Assign with 44% of the votes, but Kahoot Teach got 31% and Socrative got 25%. So there wasn't really a one platform that was really liked. And the students were happy because they gained new knowledge, and they liked that there were tricky questions, and it also helped them with revision, and it helped them to know what they need to revise more.
Also on the 16th of November, we introduced both Socrative and Kahoot platforms to math teachers at Jan Poska Gymnasium. We showed them how to log into the platforms and also explained different ways how to carry out the tests. After that, we sent them a Google Docs file with step-by-step instructions to help carry out the tests. Also, we sent them some feedback questions and received the feedback at the end of the school year. And here are the results. The teacher said that in Socrative they could see the process of taking the test and also what was difficult for the students. Also, one teacher brought to light that some teach topics differently, which caused difficulties to students because they had to think of another way to solve the problem quickly. And also they said that the tests were too long, causing the students to get tired, and sometimes there wasn't enough time to carry out tests. But overall, they said that these tests gave an overview of common mistakes and difficulties, and the students enjoyed Kahoot because it was competitive, and Socrative because students had enough time to think. That's all. Thank you.
So I guess I'll start with the opponent's questions first. Which platform is more suitable in the making of mathematical tests, in your opinion?
I think they're both great platforms, but if I had to choose one, I think I'd go with Kahoot, because there are really very many question types. As a teacher, you could make them a test, or you could ask for their opinions, or you could present with slides with new information. And also Kahoot Assign is where you'll see the questions in your mobile device and answers too. It's a great way to give homework, and as a student, I think that would be really cool.
What would be the 3 positive aspects of both platforms regarding the possibilities of creating tests?
So in both platforms, there is a question type where students have to type their answers, and I think it's great because sometimes multiple choice can be too obvious, and with typing, students have to think more of the solving part. And also in both platforms you can add pictures, whether to illustrate, or also you can add a picture of a problem, because in Socrative and Kahoot you couldn't insert equation systems, so we put them as pictures. And also Kahoot has more question types. We used puzzle, which Socrative didn't have, and Socrative has more answer opinions in multiple choice.
And if you could go back in time and start your practical research project again, what would you change?
I think I would make a plan earlier and actually stick to it, because in the beginning we talked to our supervisor. She told us that we would have about 10 questions each test, but over time it got to 20 from 25 questions. And it's actually sad because many of the teachers told us that they wouldn't do the tests because they were too long. So maybe if we had stuck to the initial plan and made about 10 to 15 questions about each test, more tests would have been carried out and more students would have benefited from it.
Thank you.
Thank you very much. I have a question. If you would be the Minister of Education in Estonia, would you use these platforms for the state examination in mathematics, and which of them?
I probably wouldn't, because there are probably some other platforms out there that have better options for making tests.
I do have a wider question. Why do you think students find mathematics hard? Is it so hard or not?
Well, it's just pure statistics. If we look at them, then it's the subject that where the most students have failed overall. So we thought it would be maybe a different approach. Maybe someone likes it better this way.
I would just like to thank you, girls. To answer your question, Mara, while listening to you, I remembered my math teacher saying, "Math isn't hard, math just needs time." So I hope with this thing you came up with, it will give students more time to spend it with math and to be a bit better at it. But thank you.
Would you like to do, in your continuous studies, would you like to have Kahoot or Socrates as a study method?
I probably would like to have them, because it's a change to the typical learning, so it would be interesting.
Do we have any more questions? If not, then thank you, Kadri. Thank you, Anette. Not the greatest. I think it's time to go outside. Raivo Jostons from Jurmala will take us on a tour around Jurmala as he tells us about history of Jurmala street names. Go ahead. Oh, you're a tall man. You can step a bit closer as well.
Mm-hmm. Okay, thank you.
That's right.
Yeah. So, hello everyone. My name is Raivo. I'm from Latvia, and I made scientific research work about my city, the Jurmala street name history. So, the topicality. There are 696 streets in Jurmala, and the exact origin of each street is not still known. So I always wondered why the exact street is called, like, for example, the Strawberry Street. Why it's Strawberry Street? Was it because there was strawberries growing or anything else? So that's why it's really interesting for me. And the purpose of the work was to get acquainted with the important part of the Jurmala's history, the street names. And my hypothesis was that the names of the Jurmala streets are related to their territorial location of the city. And the tasks were to research all the available history sources about my city, and also to group the streets by by the type. So, for example, the nature or anything else. So to group them by the types, and also to create a survey among our school students to get more information about the streets. And for this work I use descriptive questionnaires and literature and source analysis. And my, let's say, written part of the work consisted of abstract in Latvian and English content for chapters, conclusions, sources, and appendices. And also, this is the survey which I created. Maybe in this slide the most interesting question was, let's say, what was the relation to your street name with which type? So the most popular was with the place, and third more popular was with the historical person. And also another question was, is there any scene relations to your to your street name? So do you see any relations around your street which are connected to their street name? And in the conclusions, I concluded that the hypothesis of the scientific research work partially confirmed, because some of the city street names were connected to their territorial location and some were not. And the street names were given based on the most relevant, let's say, most relevant things of that time. And when I was grouping the streets, I also concluded that they were mostly connected with the nature. And I think it's because we have lots of nature in the city, and that's why the street names were mostly connected with the nature. And the street names were not given by the same principles, so it means that not only by the nature or by something else, but there were different, let's say, different principles. And pupils of Jurmo State Gymnasium do not have the sufficient knowledge about the Jurmo street names. So yeah, thank you for your attention.
Thank you. Once again, it's time for questions and comments.
Very important presentation and your work. In the title of your work, you say History of Jurmala Street Names. Did you notice some changes during the history? Because street name, as you know, ancient Romans said, nomen est omen. All kinds of names are always a battlefield of ideologies and political changes. And for example, was in the Soviet period in Jurmala Avenue of Lenin, or was this Strawberry Street since period before the World War I?
The Strawberry Street was, as I was researching always the Strawberry Street from the beginning, as I, let's say, researched it. But there were lots of streets that the names were varying. For example, in the Soviet Union, the street names were different according to their leaders. For example, the Lenin's or the Stalin's street or something like that. So they were changing a lot during the time.
And that today's names, are they, again, those that were before the World War II or before the World War I? That
changed.
Lenin and Stalin are not existent anymore, but how they were re-established, the old names, or are they given new names?
So, I'll say that they were, let's say, so, yeah, it's hard to answer. They were really changing, and they were given based on the most relevant things. For example, we have maybe some famous writers in our country, and the names were given in honor of them, for example. And if we have something maybe also popular in the Jurmala, so also the names were given based on all those other things that are important for our city. So, yeah.
So thank you for the interesting work. What's the most interesting name for you?
The first street that comes to my mind is Border Street. It's in Jurmala, and it's called, the street name is given because it divides two regions in our country. It's just the border for two regions, for the Kurland and for the Vidzeme. I don't know how it's in English, Vidzeme, but it's called Vidzeme. So, yeah. And so it's dividing two regions in our country. So that's interesting for me.
Little help. Kurseme, it's Kuramaa, Vidzeme, it's Liivimaa.
Oh, okay, thank you.
Baldies, Raivo. You were looking into the past, but what about the future? Do you have any knowledge about the naming of the streets today? What are the tendencies there? Maybe it's too far from your work, but do you know anything about that?
So, if I understood your question correctly, then do you mean that will they change or something?
Like maybe the newer parts of the city, are they named by the nature again, or maybe after some famous people as before?
Yeah, I think that by now we don't have any new regions. Let's say in our city it's just the same, it stays the same. And in each region in our city, some streets the name is given by the area in which the street is located. For example, there are around 7 areas in our city, and in each area there is a street called by that area.
Okay, thank you.
Yeah, thank you.
Do we have any more questions? Of course you do, of course you do. I'll bring a microphone as well.
How many of the 696 streets had an unknown history, the name?
I completely researched 25 streets that I know a lot about them. But other streets were still unknown for me because I couldn't find any information about them. I think because they were just not so, let's say, important. That's why there is no much information about them. But about the most important streets, I found lots of information, and also from the survey which I made among the students in our school, I also found lots of information because they told something about what they know about their streets, so it really helped also.
In Sweden we have, it's like a political decision in the district political administration that then you can find the reason for the history. Do you have the same in Latvia?
Can you repeat the question, please?
In Sweden, when you name a street, if you rename it or the first name, then there is a political debate about the names, and you get like the facts will be in the protocols of the political administration. Do you have the same system in Latvia?
I think we have something similar. It's debated a lot, and it's thought. It's not just so simple.
Thank you.
Yeah, thank you.
Do we have more questions? If not, thank you, Raivo.
Yeah, thank you.
Now it's time to hear about the district of Ida-Virumaa, where I also grew up, and Avelis Kase will give us an insight of the development of industrial settlements in Ida-Virumaa over time. The stage is yours.
Dear listeners, my name is Avelis Kase and I did my research on the development of industrial settlements in Ida-Virumaa. I studied in Poska gymnasium in 11th grade and my supervisor was Vaike Rootsma. The purpose of this research was to find out the reasons behind population changes in 4 of the settlements, all located in Ida-Virumaa. On the side you can see a map where Ida-Virumaa is brought out by the darker color. When Estonia was in the Soviet Union, a lot of Russians settled in Ida-Virumaa and it is still largely Russian-speaking. Shortly before and during the Soviet times, a lot of jobs in quarries and mines were created. So the another aim was to compare the actions taken after closing of these mines and quarries at the end of the 20th century to find out why 2 of the settlements have been left empty and another 2 are still blooming. Here you can see the settlements that I researched: Kivieli, Kohtla-Nõmme, Viivikonna and Sirgala. To help the process, I set 4 research questions and they were following. What favored the chosen settlements to transform into industrial towns and when did the population increase happen? Why are there so many industrial settlements in Ida-Virumaa? When did the population decrease start and why have 2 of the settlements been left empty over time? And what was done differently in the towns that are still blooming? Estonia produces most of its energy from oil shale. So in the theoretical part of my work, you can read about the Estonian oil shale deposit, which is one of the largest in the world, and about its mining. And I also wrote about the history of the chosen settlements. 3 of them have a long history and were first mentioned in 13th, 15th, and 16th century. But Kivieli, on the other hand, arose only in the 20th century. Fourthly, mining has had a big impact on groundwater and soil. The manufacturing of oil shale produces a lot of greenhouse gases, and another big problem is the fact that the ground has collapsed into old mining shafts. And lastly, I wrote about the current situation of the settlements. The buildings in Viivikonna and Sirgala are mainly abandoned and falling apart, while the buildings in Kivieli are all in use still, and there are only a few abandoned buildings. I compared maps from different eras and conducted an interview with a resident of Kohtla-Nõmme in April 2022. The person who I interviewed is my relative and has lived in Kohtla-Nõmme all his life. Now he's 79 years old. I chose 3 historical maps from different time periods, paid attention to the directions in which the settlements developed and to the facilities that appeared on the territory. So here you can see Kivieli in 3 different maps. The blue line represents the current border of the settlement. You can see that the land in the first map is basically inhabited. There are a couple of buildings and a railway that runs through the town. On the second map, there are a lot more houses and 2 mines have been established. The third map is the most recent. The settlement has expanded to its present borders And in the north there are 2 hills that consist of ash that was left over from oil shale processing. Both hills are over 130 meters high. There are more roads and buildings, and the mining area has also expanded. On the fourth picture you can see street view from 2011. The houses are good looking and there is no sign of abandonment. So here you can see Sirgala, which was firstly a small village in the middle of the woods. On the first map there are a couple of houses and the road goes through the village. On the second map more buildings appear. There are now 5 farm complexes without buildings and 2 farms are located outside the current borders. On the third map basically all the area is inhabited and there are blocks of flats, many private houses and more roads. Sadly, the street view shows that the buildings have been abandoned. There are no windows, the building in the back has no roof, and the grass has not been mowed for a long time. So the conclusions. The population started increasing after the establishment of underground mines and quarries, mainly due to higher employment rate. The reason behind the abundance of industrial settlements is the fact that the biggest oil shale deposit is located in the territory of Ida-Virumaa. The population started decreasing after the closing of mines and quarries because people lost their jobs and had to move elsewhere. Kiviõli and Kohtla-Nõmme have concentrated more on developing tourism, and it has played a big role in helping these settlements to keep the life going there. Inversely, nothing was done in Vivikonna and Sirgala to prevent people from leaving. The main problems in all these settlements are environmental pollution and an aging society. And for further similar researchers, I recommend to contribute more into the practical part, make interview with the settlement with a resident of each settlement, and go on spot to evaluate the situation better. And one further idea is to compare the development of settlements that I researched to a former industrial settlement from another country or state.
Thank you, Avelis. And once again, it's time for questions and comments.
So, thank you. Why did you conduct an interview with a resident from one settlement only?
It was my relative who I interviewed, so it was easy to get in contact with him. It's also difficult to get to the other settlements by public transport, and because the people who live there are old and they don't use computers, so I couldn't have made it online.
In the conclusions, you bring up that in Vivikonna and Sirgala there is a huge influence of Russian language and culture. Why have Kohtla-Nõmme and Kiviõli managed to retain their Estonian heritage to a much larger extent?
Actually, it was mentioned in the theoretical part that in Kiviõli also there are more Russians than Estonians. And the reason why Kohtla-Nõmme has managed to retain the Estonian heritage is the fact that the people there are really patriotic, and they kept speaking Estonian even in the Soviet times when all the other towns and cities switched to Russian.
And in your opinion, can focusing on tourism save settlements from being abandoned? And what else can be done to keep life going in these settlements?
Yes, I think that tourism plays a big part because it attracts people from other countries and other towns of Estonia also. And it also helps to retain the economy of the settlement. And I think that the Estonian government should start a project to make these settlements more attractive by cleaning it up and creating more jobs in these areas.
Thank you. What do you think? Is it possible to bring life back to every mine village as it was? because they were increasing in this time of this extensive Soviet heavy industry, but it is not there anymore, and maybe we just have to let them go. There are no more mines, no more mine workers, and we shall work with places which have already tourists and so on.
I think that, well, it needs a lot of money to clean up these settlements, but I believe that if the houses there are repaired and the streets are cleaned, then maybe more people will want to live there, because like the people who don't want to live in the city, it would be a perfect place for them, because it's quiet there and not much of city buzzing.
From your presentation, I got the notion that the groundwater was polluted, and there was a lot of natural. There's a lot of pollution. Wouldn't that be like? Wouldn't that dissuade a lot of people from moving there, even if there was no city buzz or any kind of noise pollution? There would still be natural pollution, and is the investment worth it? Because it would be unlike any other project Estonia has probably seen. And most of these villages are certainly extremely small, as I think you might have pointed out. So is the investment really worth it? And is it a logical conclusion to assume that Estonia should invest in Ida-Virumaa, and especially these mining villages?
Well, I agree with you that like right now, if we had more extra money, then it would be worth it. But right now, putting the money there to clean the settlements up, it's not like the main priority of Estonian government. And actually, like if you think about Kohtla-Nõmme, the water there is drinkable from the tap, because the problem has been solved, and the water there is not so polluted than in the other settlements. So I think that if you look at the still blooming areas, you can see that it's possible. But yeah, it just needs a lot of resources.
Thank you. Unfortunately, we don't have any more time, so thank you, Avelis. And now let's dive into our tie-ins. Enia Sakalovska and Ania Paula Panke from Jurvala will make us all listen when they talk about the choice of a vegetarian lifestyle in adolescence. The stage is yours.
So, hello. My name is Ania.
My name is Anni.
We are from Latvia, and we made a short presentation about our scientific research paper in nutrient education. Our supervisor is the Jurmala State Gymnasium biology teacher.
So the topic of the current research paper is the choice of vegetarian lifestyle in adolescence. The authors of the paper chose this topic because nowadays vegetarian lifestyle has become very popular, not only among adults but also among teenagers. And we were interested in examining the main reasons adolescents have chosen vegetarianism.
So aim of the research is examine the main reasons why adolescents choose vegetarian lifestyle.
Yeah, and there are objectives of the research: analyze and collect available data on the concept of vegetarianism, analyze and collect available data on the specific characteristics in adolescents, design a survey, and compile and analyze survey data on vegetarian lifestyle, and draw conclusions from research findings.
Our hypothesis is if someone in the family has vegetarian as a lifestyle, then teenager also tends to turn to vegetarianism.
Yeah, there are research methods, compilation and studies of the theoretical literature, and survey.
Structure of the work. This work paper consists of an introduction, 3 chapters, 8 subchapters, and there are 14 charts in this paper.
So vegetarianism is a type of diet based on plant-based foods, fruits, vegetables, cereals, nuts, seeds, etc., and does not include products obtained by killing animals. And properly planned vegetarian nutrition provides the organism with all the nutrients needed.
Okay. The psychological teenager period lasts from 11-12 years to 19-21 year, and this is the most complicated time in the human's life. So there you can see 2 most important questions from our survey. In the third chart you can see the question was, are you a vegetarian? And as you can see, 84% answered that they are not, but 50% answered that they were vegetarians.
And the question in the fourth chart was, why do you choose a vegetarian as a lifestyle? And 41.2% chose it because they wanted to try it, 23.5% because there's a vegetarian in the family, 29.4% for other reasons, and 5.9% because this lifestyle is popular these days.
Conclusions. Our hypothesis was partly correct. The study showed that only 23.5% of teens turned to vegetarianism because their family has this lifestyle. Yeah. Thank you for your attention.
And once again, it's time for questions and comments.
Thank you. I have children. They have friends, and it's quite popular to be vegan, not vegetarian, but vegan. What is how it is in Latvia?
Vegans.
Vegans.
Yeah.
Maybe this vegetarian is not so popular at all, or maybe
I think
there is a need for a next study about vegan.
Well, no, we don't. No, we won't do it.
Thank you.
Thank you for the work. I got myself thinking, do you have any insight why people quit the lifestyle?
Why they quit?
Yeah.
I think maybe sometimes it's hard, or maybe they like meat or something like that.
Maybe it's hard for them to not to do it if you have done it for like years, and then you stop it, and then it's hard for you to go back to the normal lifestyle.
Thank you, girls. Thank you. It seems quite an interesting topic, especially in a nowadays world. When you did your research, did you come up? Do you have any opinion, or what are your thoughts on? Are the young people well informed before making this decision? Do they know which products to consume, how to add up all the vitamins, and how to be healthy during this change of
I
status?
think they just Google, and I don't think they know all the things how you should be, and you just.
But does it look like they do it not the right way, but are they like well informed? Yeah, yeah.
I think some of them are really informed. They check all information, but there are a lot of people who do it just don't know
just to try
information,
it,
yes, and don't check all the information, yes.
So are you thinking about publishing your work to draw more attention to the...
No,
do you?
No.
I think you really should. But yeah, thank you.
Do we have questions?
Are you vegetarians yourself?
No. Well, she's kind of. She's kind of. She don't eat meatballs, but if it's in a burger, then she eats. She's like kind of, but not at all.
But not.
Do we have more questions? Seems like we don't. Thank you, Enya. Thank you, Anja. Waste management has become a pretty hot topic recently. Helerin Peterson from Jaan Poska Gymnasium will introduce us to the topic as she speaks about an overview of the Estonian waste management and the waste sorting habits of the residents of Kastre Rural Municipality. The stage is yours.
Hello, everybody. So the aims of this research were to learn about general waste management in Estonia and also how the waste is processed after collecting it. I also studied the waste sorting habits of the residents of Kastre Rural Municipality, and I wanted to know what the residents think should be changed in the waste collection system of the municipality. And on this map, you can see that the red territory is of the municipality of Kastre, and the little blue dot marks Tartu city, so you can see the difference in size. So in Estonia, in Estonian waste management, most tasks are performed by the municipalities, and there are 79 of them. And in Estonia, after collecting the waste, a lot of it is exported to the countries who can recycle it. And it is also incinerated in big quantities because mixed municipal waste actually has almost the same energetic value as oil shale, which is mainly used to produce electricity and warmth in Estonia. Package waste is one of the main types of waste, and in Estonia there are 3 package organizations who deal with the collection of waste. So they provide the municipalities with package containers. And here you can also see the logos of the companies. Kastre Rural Municipality has 2 boroughs and 49 villages, and there are about 6,000 residents and 2,500 households. In the municipality, there are 35 package containers provided by the same 3 organizations that I mentioned. And on the map, you can see all the containers. This map is actually also on the website of the municipality that was recently uploaded, which is very good news. And all the yellow dots are for mixed package. And then there are also some paper and cardboard package containers and 1 glass package container. So I set myself 5 research questions, and I wanted to know how many people actually sort out all 3 main types of waste: biodegradable waste, waste paper, and packaging waste. Then if they don't sort them out, then why? And which is the most sorted out types of waste. I also wanted to know how many residents compose their own biodegradable waste, and of course what the residents of the municipality think should be changed in the waste collection system. So in order to answer the questions, I made a questionnaire in Google Forms, and I worked together with the municipality council. So they shared the questionnaire in the official Facebook group of the municipality, and then after the study, I shared the results with them. And there were 11 multiple choice questions, so the answering would be quick and easy. And I received answers from 197 households, which is about 8% of the households. So what were the results? I found out that mostly sorted types of waste are pledge packaging, bulky waste, electronic waste, and hazardous waste. And on this diagram, you can see all the other types of waste too, how much they're sorted out. Those are the types of waste that are usually produced in households. And more than half of the respondents composed their biodegradable waste. Then more than 80% of the respondents sort out all 3 main types of waste. But there are still several problems with the waste collection system, such as the shortage of containers, insufficient emptying frequencies, and too small container sizes. In conclusion, not all residents of the municipality sort their municipal waste, even though it is mandatory in Estonia. And most respondents are not satisfied with the waste collection system because there are many issues, and they mainly regard containers of waste paper and packaging. And there still is a need for an opportunity to give away biodegradable waste because even though it's like in the countryside, then there are some people who live in apartments, in blocks of flats, and they don't have the opportunity to compost. And now this is all from me. I'm ready for opponents' questions.
Thank you, Helerin. And yeah. And you're already going to know what I'm going to say. Time for questions and comments.
So, has the author already presented the results of the research to the Kastra Municipality Council? And has the council planned or is ready to plan changes in waste collection system based on the results of the survey?
Yes, I sent them the results about a month ago, but I still haven't got an answer. I don't know if they have read through the study, but they were very interested in the results because then they would have like some kind of a picture of the situation in all the villages and boroughs.
So thank you for that. So thank you for the work. I was wondering, do you have any good examples in Estonia where the waste management is really good? Or not, if
well,
not in Estonia, maybe abroad somewhere?
Well, I can talk what is good in Estonia. it can be improved still, but for example, waste paper is one of the types of waste that is like mostly recycled, like recycled to most compared to the other types of waste. For example, about 30% of waste paper is recycled in Estonia in paper manufacture in Rapina. And I mean, they make new paper and other paper products of the waste paper. Mostly they make those like cardboard corners that people use to protect products when transporting them.
Thank you. I have a question about your respondents. What about those people who do not use Facebook? Mostly elderly people.
Mm-hmm.
And they maybe belong to that group who was saying we have absolutely no waste.
There were some people who said that, oh, we don't have waste. I think maybe they meant that, you know, the little waste they have, they just put in their own personal container. And then there's a waste company that empties their containers regularly. Then, what was the first part of the question?
You had only or it's quite many, about 200 answers, but it was 8% of the households.
Mm.
But what about those people who are not using Facebook? How to reach them?
Well, at first I thought I was going to go door to door and ask everyone orally, because then I would get also answers from older people, but that would be too time-consuming, and unfortunately there isn't really another way to reach the elderly. And I really think that putting the questionnaire up in Facebook was, in this case, better, because I did quite many answers.
Yes, thank you. And that would be maybe a task for the municipality to do the next step.
Yes.
Thank you.
Do we have questions from audience? Of course you do, of course you do.
This is a hypothetical question, but if you look into the future, we are going to have to pay for waste.
What?
We
I'm
are
sorry.
going to have, if you look into the future, it's already coming in Sweden that you will have to pay for your waste. You have to pay to get them to the dumps or in the waste bins. You have to pay money for it. What do you think the people will say about that, and how do you think they will react?
Well, right now we have to pay to get our waste to the landfills. Even if we take our mixed municipal waste to the waste station, for example in Tartu, then there is a price for that.
And if you turn it around, what do you think people will think if they get paid for the waste?
Why? I'm sorry, I don't hear you.
If people will get paid for the waste, because waste is going to be money in the future.
Oh, like, okay.
What do you think they will say? Will they use it more?
Yeah. I think the world is like going towards that kind of world where waste is actually seen as a resource more than something that has to be gotten rid of. And yes, I think people will start using it more.
Do we have more questions? Seems like not. Thank you, Helerin. So it's time to dive into physics. Sophie Malmgren from Tibble will go into details as she presents us the topic of the basic characterization of nanomaterials. The stage is yours.
Hello, my name is Sophie, and I got the opportunity to investigate the basic characterization of nanomaterials at the Stockholm University together with 8 other students from around Stockholm. Oh, yeah. Okay, so the aim of the report was to explain the basic characterization of nanomaterials. And in this project, cellulose nanocrystals, silver, gold, zinc, and iron were chosen to be investigated. However, in this report, only cellulose nanocrystals and silver nanoparticles will be researched about. So a variety of methods were selected to characterize the different nanoparticles. So we have the dynamic light scattering, showed the size distribution of the particles regarding its intensity, numbers, and the volume taken per size class. And then we have the image from the scanning electron microscopy, and shows both the formation of the particles and the morphology, and can also be used to determine which elements are occurring in the different objects. And this information can also be collected by analyzing the energy from the X-rays broadcasted from the objects. And then the stability of the nanoparticles can be determined through the zeta potential and is measured to enable a comparison with other materials. And then by comparing the outcome with former results of the UV, the values makes it feasible to settle which element is being researched. And the range we used was between 300 and 800 nanometers. Yeah. So the presentation or the preparations of samples for dynamic light scattering was to dissolve the particles in water because of its transparency. And for the UV, the solution, if the solution is solid, the light will be reflected and not absorbed. So to create the best conditions, the nanoparticles should be solved in a liquid. And then for SEM, the preparations conclude dissolving the particles in water as well. And then a drop of the wished solution will be put on a metal holder. And then when the water is evaporated, a thin layer of the solution will persist. And the remaining solution will then be observed in the electron microscope. So for the results, you see on the left side, figure 1. And there you can see 2 different peaks, where the first one is at 23 nanometers, and this has a 4.6% intensity. And then the second peak by 196 nanometers and has a 95.4% intensity. And then in figure 2 on the right, only one peak can be seen on the diagram, showing the number for CNC, and the peak is by 23 nanometers as well. And then in figure 3 on the left, you can see the zeta potential for CNC, and it can be observed at negative 51.1 millivolt. And then for the UV on the right, the absorptions of the different wavelengths was measured to around between 300 and 500 nanometers. And then for silver, we start off with the UV, and the absorption of the different wavelengths was measured to around 400 nanometers. And then we have the dynamic light scattering, and in figure 6, the size distribution by intensity. 3 different peaks can be observed, and the first one at 0.82 nanometers, and the second one at 2.25 nanometers, and the third by 19.63 nanometers. And in figure 7 on the left, you can see the size distribution, and it can be seen at 0.82 nanometers. And then on the right we have the zeta potential, and it can be observed to negative 44 millivolt. Yeah, our findings on the basic characterization of particles by using different methods to extract information on several properties of different materials were compared to public research and were in line with the previous published data. And UV can be used for studying the optical properties of nanomaterials in dispersion, and the DLS can be used to take the hydrodynamic size and size distribution of nanoparticles of a certain sample. And the SEM illustrates the nanoparticles morphology. Yeah, thank you.
For the last time in this session, it's time for questions and comments.
Tack, Sofia, for your really interesting presentation. I have a question about the bigger picture here, or like, can we now use the nanoparticles thanks to your research and new information? Can we now use it for something new in some other area, or?
I'm not really sure, but I think we're about to evolve, and in the future we may be able to develop the medicine with the help of nanomaterials and the different particles. Yeah.
Okay, yeah, thank you.
Thank you for the work. I believe that nanomaterials can be used on drug delivery in the future. I read an article once about it, so not to brag, but just to give an example. Do you know what are some of the downsides of nanoparticles or nanomaterials?
Maybe if they're let out, they're so small that we may not be able to filter it. So if it gets in the water, and when we then are going to collect the water, they are so small that they will not be filtered away before we drink it. Yeah.
Do we have questions from the audience? You don't have a question. Wow. Okay, no one wants to take a word? No? Thank you, Sophie. So I want to say thank you to all the presenters of the second session, but now it's the most important time of the day. It's finally time for the long-awaited lunch break. To our Latvian and the Swedish guests, please bring your lunch tokens with you, as it was yesterday, and we'll meet again in this room at 13:40. Thank you.
Nii, teen heliproovi, kas sa kuuled mind? Teen heliproovi, kas sa kuuled mind? Mina mingit mürinat kuulen, nii et on kuulda auditooriumit. See tuli mulle nüüd umbes 20 sekundit hiljem tagasi minu enda hääl. See tuli mulle 20 sekundit tagasi minu enda hääl seal koridoris. Mina teid ei kuulnud rohkem, aga ma kuulsin enda hääl. Kas me nii ei saa teha, et me paneme selle arvuti taha selle suure selme? Anna see Peeter, anna see juhe siia taha.